Hello, my name is Rob Gordon. I am a senior health science major, and the premise of my research is to understand the efficacy of clinical application for sequencing the 16S rRNA bacterial gene in healthy Colby-Sawyer students. My research problem is that there's limited research about how genetics and social determinants, such as oral habits, influence the types of bacteria found within the human oral cavity. There's also limited research on the most effective way to collect bacteria, as well as the proper way to assign correlations to the results found. This method of DNA analysis is fairly new to science, and proper taxonomy of bacteria, species, and genuses has not seen consistent enough correlations to draw conclusions on the effectiveness of this analysis type. My first research question is, how does genotypic analyzation of bacteria in the oral microbiome allow correlations to be made between demographics and oral health of a subpopulation? Another question is if sequencing the 16S rRNA bacterial gene is an accurate and precise method of bacterial identification and quantification, and if true, can this method be used in a clinical setting? The hypotheses of this study are that there will be correlations drawn between demographics and oral health of a subpopulation, and that sequencing the 16S rRNA bacterial gene has direct clinical applications in such fields as dentistry. The proposed participants of this study are 18 to 25-year-old Colby-Sawyer students. For the proposed methodology section of my study, students who qualify for my study are students of Colby-Sawyer who are between the ages of 18 and 25 years old, do not have existing medical conditions that could put me at risk, and do not have temporomandibular disorders such as TMD. For recruitment of this study, students will, in early January, be recruited via email and class recruitment during classes taught by Ivy science professors. Students will understand through a brief description of what my study is during class recruitment periods and have a place to sign up for more information. Students will be asked to sign up for one meeting where they will complete a demographic survey and undergo saliva sample collections. After the demographic survey is finished by the participant, I will begin obtaining saliva samples via the drool method. I will ask you to rinse your mouth with water to eliminate any food debris and swallow the remaining saliva. I will ask you to remain silent, to sit upright on the edge of the chair, keep your mouth open, and tilt your head slightly downward so that the saliva can passively drain from the oral cavity and lower lip into a sterile collection device. I will continue to have you passively drool into a cup until about 20 milliliters of saliva DNA has been collected. Collected samples and surveys will then be labeled. Samples will be placed in a freezer until further analysis. The samples will then undergo isolation for the 16S gene through a series of enzymes, primers, and washes. The samples will then be analyzed to determine the quantity and concentration of the extracted samples. The samples will then undergo replication via PCR replication to amplify and make millions of copies of the 16S gene extracted. The samples will then be cleaned up via PCR cleanup, which is a process that removes unwanted components such as the primers, enzymes, and buffers used in the previous steps. The samples will then be prepared for sequencing and sent to UNH's genetic laboratories for analyzation of our samples. The data we get back from UNH will be in the source of a computer file that will read all the base pairs found in our sequenced samples. The data will then be uploaded into an rRNA database that will name, quantify, and graph our sequence data. During our initial meeting, where saliva samples are collected, prior to the collection, a survey will be given to the participant that is looking to learn how demographic data of your genetics, environmental factors, and oral habits influence the composition of oral bacteria found within the oral microbiome.